visualizations that were produced by a collective of scientists as well as designers with the purpose of really understanding complexity and understanding networks. I'm a network scientist, meaning that I study various complex systems, the cell, the universe, the social systems, economic systems, and try to understand the connections between the pieces in those systems. And part of our scientific practice, we also developed a whole line of research and thinking about how do we visualize these networks. And we did so not for the primary purpose initially to be in the museum with them, but mainly to understand these systems, to really understand complexity, to be able to see that. And what you see here at this exhibit is a selection from those visualizations that we have done. I like to start the exhibit at this particular image, which in a way dates my interest in network science as well as our interest in network visualization. I know exactly when, for the first time, the concept of network occurred to me. I was working at IBM right after my PhD, and for Christmas I took home a book. The book focused on computer science, and within that book I found a little article focusing on computer networks and networks as a mathematical object. At that time I was living in New York City, and made me realize how many networks make the city work. The communication network, at that time the internet was just emerging, phone lines and so on, and I realized there's so little understanding of these complex networks around us. So I decided at that moment, during that Christmas break, that I will focus on networks, to understand complex networks. And what is interesting is that the first product of this thinking was not a research paper. It took several years for me to publish a paper, but rather this particular visualization. This image is also very well dated because we have the original print from the IBM computer that dates it as 1995, January 17, which implies that 3 weeks after I started thinking about networks, my first step was to visualize. What did I visualize? In this case, I visualized the network of water as it enters the ground that is common, for example, in oil exploration. And in particular, I visualized the mathematical model that describes that particular process. And what is interesting from a visual perspective, that this particular image kind of aesthetic look is very deeply rooted in my training as a physicist. I show a network as a lattice with only straight lines corresponding to the links, and the nodes don't yet appear. And we will see that with time, that particular representation drastically changes. But nevertheless, this is where our journey begins. As I mentioned, the first visual language was that of a square lattice in 1995. But in 2000, 5 years later, by which time we started to publish on networks, the whole visual language changes. Now you see explicitly the nodes, and the nodes are colored. That is, the node color carries information, and they are connected so far with straight lines. And the 2000 image is still depicting a model network, in this particular case, the so-called random network. But soon the year after, in 2001, we start showing real networks. And that change becomes a hallmark of the lab's visualization, because what you see in the 2001 image is already the protein-protein interactions based on experimental data and how the proteins are connected within our cells. In 2004, we are faced with another challenge. We need to have information about the links themselves, so we start coloring the links to show the fluxes that flow, in this case in the metabolic network. And in 2007 is the first time that we start encoding information into the node size as well. And this came in the context of human disease network. 2011, we encounter very dense networks where the straight lines don't actually do it any longer because it's too confusing. So we start developing algorithms to curve the links to avoid the dense areas. And 2018 is another major change in the way we represent networks, because for the first time we are able to step out of 2 dimensions and enter the 3rd dimension. That is to really start printing 3D data sculptures. a year later, in 2019, the technology emerges to do that in color as well. That is to encode information into data structure by coloring the nodes. And in 2020, we encounter so complex networks that it's impossible to really even print it, and those are shown in virtual reality. About 3 years ago, I got a very unexpected request from the journal Nature. As many of you know, Nature is one of the most influential scientific magazines out there, and the editors were getting ready to celebrate the 150th anniversary of the journal. And they came to me and asked me if I could help them design the cover for the anniversary issue. This was an unexpected request in a sense that it was the first commission we ever got to visualize something for the sake of visualization. And we approached it from a data perspective. We started by first mapping out about the 80,000 papers that Nature published in the last 150 years. And then we were looking for hidden connections between them. That is, in this image, what you see behind me is all the papers that Nature has published shown as individual nodes. The colors correspond to what disciplines we have: physics, biology, social sciences, chemistry, ecology, and so on. But the links are not links that would be obvious if you read the paper. The papers were connected if they are connected in the scientific community's mind, if in the mind of the researchers there is a connection between them. And how do we extract that hidden knowledge? We did so by finding how often two papers were cited together in the scientific literature. Because the more often two papers are cited together, the more related they are in the minds of the community. Hence, on the map, you see regions of similar color, like yellow color, blue regions, and so on. And that's because physics papers tend to be cited together, and biology papers tend to be cited together. But you see also papers that have different color and they're connected, because those are what we call interdisciplinary or cross-disciplinary papers that multiple communities find it to be interesting. In many ways, this image shows the extreme complexity and heterogeneity of science itself, the existence of the multiple fields. But it also shows that they're not disparate disciplines. There is a conversation between the disciplines, and those are captured by these hidden connections that we have between the different papers and different disciplines. The project resulted in the Nature cover that is shown here next to me. But at the end, we had so many interesting visualizations that the one-page request to design a one-page cover turned into a five-page cover that is a fold-out right now, showing first the map itself, and in the back of the fold-out, we also showed the impact of the individual research papers, where they were inspired for. They're shown as a carrot, and also what kind of research did they inspire. What is interesting about our visualization journey is that a few images have been published, either as the cover of a journal or they are inside the research paper. But they are accompanied by many sketches, as I would call them, or many trials. And I also want to emphasize that for us, visualization is not only for the final product, but also to understand the systems that we're studying. What you see next to me on the left and the right are those sketches that preceded the final product. And what you may recognize is a kind of a black and white version of the final Nature cover, where we're trying to work on different type of layouts. And while these images all look alike, or at least 3 of them look alike, if you look closely, you will realize that there are very fine differences between them. And those fine differences are really looked at when we're trying to decide what should be the final layout. The one on the other side is also one of my absolute favorites. It's a protagonist to the carrot image that ended up in the back of the Nature cover. And we ended up not using this particular representation, but I thought visually this was really stunning. So I really wanted to have them in the museum exhibit. And it's also important because we use these images to show how much in the lab in the last 25 years have worked commercially for our drawer. that we created hundreds and I would say thousands of images that we knew that we will not be able to show. Nevertheless, they were a very integral part of the journey. What you see here is the universe as a network. Galaxies connected to each other through the gravitational forces that hold them together. And this is a beautiful representation of the collaboration the designers and the scientists have in my lab. These images were generated by Kim Albert, who is a German-born designer who came and worked for 2 years in my lab, and now he's back to Berlin, where he continues to work in design. And when he joined my lab, he was embedded in a group of physicists and astronomers, because at that time we were interested in trying to figure out how do we think of the universe as a network. And the result of that journey was a research paper in which Kim himself was a co-author as the designer, because it was so much based on the visualizations. But obviously there were lots of other scientific results as well. In many ways, the images that were created from that, the video that you see in this exhibit, as well as the prints, offer a stunning representation of the cosmic web. Because we tend to think that the stars and the galaxies are lonely objects. In reality, they are not. They are connected by gravitational forces, and the evolution of the universe is determined by this cosmic web. And we wanted to bring this cosmic web back to us so that we can feel it, we can understand it. What you see on this map is what we call the flavor network. And when we generated that, we were trying to answer a very simple question about food. What are the ingredients that taste well together? That is, what are the ingredients that we should really mix in our food? And we were inspired by a hypothesis from molecular cuisine that said that two ingredients taste well together if they share the same flavor chemical. It's a little bit like dressing up and matching certain colors so that we have some consistency there. So therefore, we created a network where the notes are the different ingredients in our food, and they are connected only if they share not one but several flavor chemicals. That is, they have a similar flavor components. For example, you see there's a connection between cocoa and coffee because they share multiple chemicals that defines their taste. Now, the question is, what did this map tell us about what tastes well together? And in a way, it said that the molecular cuisine hypothesis is right and wrong depending on where you look. Turns out that the Western cuisine really prefers to combine ingredients that have many chemicals together, but the Eastern cuisine is combining complementary ingredients, those that don't share flavor chemicals. This map was also the one where we first had to curve the links because it was too dense for us to be able to see how the different pieces are connected to each other. And particularly if you look in this region, you see that there is a cacophony of links that is very difficult to disentangle. So I want you to kind of remember that because we have another solution to this problem. Indeed, because mathematically we know that in 2 dimensions, we can never represent a network without the links crossing each other. But we can do so in 3 dimensions. Let's see how do we do that. The network you see here is identical with the flavor network we just saw earlier. The major difference is that that was in 2 dimensions and this was in 3 dimensions. Now, why 3 dimensions? Well, on one end, before I became a scientist, I was studying to become a sculptor. And thanks to that experience, I was always frustrated that network visualization is stuck in 2 dimensions. We produce images for the screen, for a piece of paper, and not really showing them in 3D. On the other hand, there's a mathematical theorem to tell us that any network can be laid out in 3 dimensions without the links having to cross each other, versus in 2 dimensions where they must cross. So therefore, since 2007, I've been trying to find a technology through which we can bring the networks out from the computer screen. We succeeded around 2017 and 2018 when 3D printing emerged to the level that we could actually start systematically print networks. Now, looking at the 3D version of the flavor network, you realize how much more content has emerged that you could not do in 2 dimensions. But the most important question is that how do we lay it out? How do we decide where the nodes and the links are? And in order to do so, we have to build algorithms, or in other terms, we need to formulate the aesthetic problem of a good network layout in terms of the mathematics of the problem that the computer can understand. And hence, the principle that we use are summarized in this equation. So this is the equation that the computer sees, and the way it does it is shown actually in this video. It solves this equation as it's searching for the layout that really captures the best the structure of the underlying network. And you can see it unfolding and finding the final structure will end up actually in this particular structure. What is being encoded in that equation? What we encode is that we want the nodes who are densely connected to each other to be next to each other. That is, the communities to correspond to nodes that are close to each other, and those who are not connected to each other could be far away from each other. So when you look at this network, it really reveals the underlying structure of the system. And now we also have a mathematical methodology as well as a technology by which we can take arbitrary networks and lay them out in 3 dimensions and give a very different experience and different way of interacting with the network itself. One of the problems with the 3-dimensional sculpture is the information about the nodes and links is lost now because we cannot color the nodes easily. That information is given back to us with augmented reality. So if you come to the exhibition space and you get this iPad, then the iPad now will reveal the identity of the nodes. So for example, it will tell you that that big central node over there corresponds to cocoa, and next to it there's rose and so on, and it will tell you that these are mostly cheeses, and these correspond to mostly fish, different type of fish, and down here we have the fruits. So augmented reality gives a possibility for us to regain the content that is lost by simply looking at the structure of the network, but the structure itself now is much more revealing than what we could get in 2 dimensions. The possibility to bring back the content is thanks to MOME, the university in Budapest, and several creators who worked with them, who created this interactive feature that we can regain the contact from any 3D sculpture and kind of reveal again what are the nodes and what are the links. These are researchers and designers from the Moholy-Nagy University in Budapest who have worked tirelessly to make this possible for us. The 3D printed networks are also interesting us for another reason, because they're examples of physical networks. And why? Physical networks are networks where the nodes and the links have physical existence. And we're interested in them because they mimic real systems in which physical networks emerge, like the brain. What you see on my left and on the right-hand side is a neural map of the mouse brain, of the same mouse brain, as a matter of fact, one as a light box and the other case as a video. And what it illustrates is the dense environments in which the neurons exist. So the difference, say, between the Nature 150 work and this one is that in many networks, the links are virtual. They just represent some association between the links. In these cases, the links themselves, the neuron, take up space and they push against each other. And that physicality determines the structure of the network. So one of the big research projects that we have in the lab is to understand physical networks. And this is all motivated by the brain as a network. We hope this understanding will help us understand how the brain has emerged and how it functions. The 3D networks that we saw earlier corresponds to only one limit of a physical network, or one phase, as we like to say. And in a counterintuitive fashion, this fat gurkha, as we call it, is really a physical network, but corresponds to the other limit of it. The way we should think about it is that the statue that we saw earlier is like water, and this is ice. And here what we're showing what happens if a network becomes more and more crowded. As the links are increasing in thickness, there is less space for them to find their location, and they must push against each other. And then you end up in this very stressed state that we call it the gorge or the gurgas, the Latin word for gorge, because really there's no space for the links to find their destination. Why is this state important? Because this is the state that really describes our brain. We tend to think of the brain as being a network where everything is connected to what they should connect to. But in reality, in the brain, things are only connected to what they can, because they're in a very dense environment, the neurons themselves. And mathematically, that dense environment is described by this Gurka object. This is a static image of that, and what you see on the video is how do these Gurkas emerge? How you go from a very thin network where there's lots of space between the nodes and the links to the very stressed and dense network state. In physical networks, the links don't have the luxury to go straight to their destination. They must curve to avoid each other. So for several years, we were working in the lab how to describe this curving process. How do we characterize and distinguish a curving link from a straight one? And hence we introduced the concept of the link temperature, the network temperature. That just like in the air, if the air is hot, the molecules move very fast. And as it cools down, they move slower and slower. We ended up assigning high temperature to the links that need to curve very much and low that go straight. But we didn't have a good way of representing that. And in 2019, in the summer, I was in Vienna at the MUMOC to see an exhibit about op art, and I saw the many, many square lattices the artists were using, and that inspired me to really think of the heat as a way of represented by a square lattice. So what we did in this particular image is that we changed the temperature of the links by making it very hot in the middle, hence the links are very curvy, and decreasing it towards the edge of the lattice. And that generated this amazing final representation that is, by the way, on the cover of the catalog of this exhibition. How does an idea become an artwork in the museum? And as I mentioned, it was in MUMOC when I got inspired by the lattices, and then I went back to Budapest and I said, how could we show this hot network? And I ended up drawing this. I draw lines that the temperature is increasing, then I draw a square lattice where the center is increasing and becomes hotter and hotter. And then I wrote an email that you see over there together with the drawings, and I sent it to Yan-Chen Liu, my student in Boston who was working on the hot lattices. And I asked him, could you implement algorithmically this increasing temperature towards the center of the network and show me what you got? And when I got about a week later the image, I was totally impressed. That is the image that we saw earlier, which is now on the cover of the catalog. So when the COVID came, we were wondering how could we create a work that really represents this moment. So we ended up getting data from New York City of mobile phone information, how many people are in the city, and we used the long analog where the map of the city was expanding the more people can be found at that given hour within the town. And we used the long analog because my lab has been studying a lot lung disease. And you see that during the day, they expand and then it starts shrinking. But it should not escape anyone that we're talking about March 14 over here. So this is the time when COVID was already present in New York, but people were not yet used to it. But then the week after, you actually start seeing that fewer and fewer people are coming to the city, even though they could. And hence the lung is not expanding as far. It is not such an expanded breathing. And then of course comes March 21st, when effectively the city is shut down and the lungs are collapsing. What is amazing for me is not the fact that when the city shut down, the whole lung, the city collapsed, but the emergence of a new normal. So New York City in the coming weeks continues to breathe. During the day, people come in, workers come in to do the essential services, and then during the evening they leave. But it does so as a sick lung because the amplitude of the motion is much more reduced. I call this work my 21st century because I really do believe that March 21st, when New York City has a lung collapse, really marks the beginning of the 21st century. In the same way that it's hard to remember what happened in the 20th century century before the First World War, I believe that generations later we will look back at the COVID moment of March 2020 as the beginning of the 21st century. What you see here is what we call the human disease network. And the origin of this map goes back to me returning to Boston in 2005 and joining the Dana-Farber Cancer Institute. And 2005 was a few years after the Human Genome Project. All my colleagues at Harvard Medical School were discovering new disease and gene associations, that they were discovering almost on a weekly basis genes that cause certain diseases. And what I became curious is that how do we conceptualize all of these independent discoveries? And hence we built this map where each node corresponds to a disease, and the size of the node corresponds to how many genes were known to be associated with that. So you see deafness as the biggest node here, because about 33 different genes were associated with that. The colors of the node corresponds to different classes of diseases. For example, here you have mostly cancers in blue, and two diseases are connected to each other if the same gene is responsible for both of them. So for example, when you look at diabetes and obesity, these are very related diseases, and they have multiple genes that kind of link them together. In some way, this image was the lab's most reproduced image historically, because for the first time around, both researchers as well as those interested in medicine to really see the full landscape of diseases and the connectedness. And it showed us that diseases for which but to go to different hospitals at the genetic level are connected to each other. Originally, it was published as an image in a figure in the National Academy of Sciences, but the National Academy also carried a poster with the paper that became very popular. And then the New York Times carried a full-page version of this image. But it was also this map that really started our conversation with the art space, because Hans-Ulrich Orbist from the Serpentine Gallery has found this image and the flavor network and exhibited it at the Serpentine and eventually put it in the catalog. And it was the first time really that one of our maps really emerged in the art space. What you see here is a brand new project. In 2021, we were asked by Nature to again work together, and this time to create a cover for the 20th anniversary of the Human Genome Project, the project that inspired the Human Disease Network. So I was delighted to work with them again because I felt like we came full circle. And what you see here in this room is the ability to see how you can represent the same data in many, many different ways. What ended up on the cover is this representation, an iceberg analogy, where what you see as circles, and you see it particularly well here, each circle corresponds to a chromosome in the human genome. And the circles and the smaller circles inside the big circle correspond to the individual gene on that particular chromosome. But there's another layer of information encoded onto that, which is how much research has focused on that particular gene. So if particularly here you see the p53 gene, this is the so-called cancer gene, an extraordinary number of papers have focused on that, and the size of the circle corresponds to how much research has really been focused on that particular gene only. You can see from this image that a few genes got exceptional amount of attention, but most of them have been really ignored by the research community. This is the representation of the same idea, except that the iceberg now encodes two different types of information. What you see above the waterline is how much was known about the gene before the Human Genome Project. And what you see under the waterline is the new knowledge that the Human Genome Project brought to us, that how much research has focused on those genes after they have been mapped out, in a way illustrating how much was under the water that at that time was unknown to us. This image encodes exactly the same information as the iceberg one that we just saw a second ago. But now the impact of each gene corresponds to different buildings, so we chose a city landscape to represent it. And the height of the building corresponds to how much work has been done after the Human Genome Project, and the shadow corresponds to how much was known before. So if you look carefully, you would realize that not all buildings have a shadow proportional to their height, because the amount of interest often before and after the Human Genome Project was different. And on the left-hand side, you see again the same data being represented. But now we used, instead of a city or instead of an iceberg analogy, one of a cell's nucleus, where we crumpled the genome, those circles that we saw earlier, inside a sphere, kind of mimicking the fact that the genome itself is deeply crumpled inside the nucleus. And to encode information about how much interest is about the particular genes, the thickness of the line, the thickness of the genome itself is varying to encode the amount of research focused on that particular region of the genome. The colors correspond to different chromosomes. And in a way, what I find totally amazing about these three images is that how many different analogies and how many different language and vocabulary you can develop to show the same data. And what is common between them is the accuracy of the data. That is, we have a fidelity to original data, but we have a very, very different visual look that we generated for them. So when COVID came, my lab got deeply engaged on trying to find drugs to cure COVID patients. And the goal was really not as much to design a new drug, because a new drug takes often a decade or two to really be introduced to patients, and there was no time for that. But rather to find out whether any of the 6,000 already existing drugs on the market could really help cure patients with COVID-19. And for that, we developed a whole set of network medicine-based tools that looked at every single drug and said, could it potentially be effective against COVID-19 by seeing what are the targets of the drug. And through that process, we predicted a series of drugs that could be effective, and then we experimentally tested that. This project over here is generated to show that extreme search process that we have to go through to identify the few drugs that may be effective. If you come and interact with it, this is an interactive installation, you will have the option to really select a particular drug on the screen. And once you do that, then the next image, once you have kind of selected, and here I'm going to select newly another drug over there by clicking on it. And once you achieve that, then on the second screen, we will actually get to see where are the targets in the large network of the human protein interactions of that particular drug. And then on the other screens, we show the evidence whether this should or should not work for COVID-19. And in particular, on the bottom screen, the red screen, what we see is that not only we theoretically predicted what should work using the network, but also our colleagues at Boston University ended up experimentally testing each of them. And in that particular image, when you see a blue region, it means that there is a cell there. But if it's surrounded by a red region, it means that inside the cell there are COVID viruses, so that cell has been infected. And those where you are lacking the red region, it means that on that region, that particular drug did manage to kill many of the viruses. In this particular case, the molecule we look at it was somewhat effective to kill the viruses, because there are big regions of the plate where the virus is missing. We ended up representing this whole research project, that desperate search project for a drug, in these particular videos, but they also, the successful finds are shown as glass boxes over there. We have about 10 boxes, each of them corresponding to a different drug, and the network within the cell that allowed to correctly predict the effectiveness of that particular drug. The way I think about the data and the visualizations is to kind of create multiple sketches to represent the ideas. And this wall shows a subset of the many sketches that I created in the last few years that I use for multiple purposes. I use to record inspirations from artists that I see, or even from nature or from science. I also use it to offer feedback to my lab and my colleagues as they process certain images. I use sketches to design certain new situations and new artworks, and I use them also to capture ideas about the journey in the art space. And I was delighted to see that the curators decided to put a selection of these sketches over here. If you look carefully among these images, you will recognize the predecessors of many of the projects that we see, or you will see process images as they evolved, arriving to the shape that are shown in this particular exhibition. In 2018, we decided to take the tool set of network science to really reflect on art and the organization of art. Why would we do that? Because I believe that really art and the value particularly in art is generated through a network. That is, whether a certain object is valuable or not, it depends on who touched that object. Where do you see that? What museum? Is the prestigious museum or not so prestigious gallery? What did the artist do before and what did he do after? But the question is that how does this value really emerge? And in order to understand that, we ended up mapping out the history, the exhibition history of about 500,000 artists. And from that, we generated a network of institutions. And the network that you see here is the result of that project where each node corresponds to an institution. The size of the node corresponds to the prestige of the institution. So here you see MoMA, Guggenheim Museum, Pace Gallery, and many of the top European institutions, including ZKM. And two institutions are connected if they tend to exhibit the same artist. Why is that link meaningful? Because if two institutions exhibit the same artist, like the BarabásiLab has been exhibited at the Ludwig Museum and now at ZKM, it shows that the two institutions have a different conception of what contemporary art really means and what is value in art. This network was very unexpected for us because on one end, of course, showed that the high-profile institutions all form one cluster, which means that they tend to exhibit the same artist. They're fighting for the same artist. But it also showed the existence of multiple communities of institutions, often geographically organized. Here you see Eastern European institutions, and here you see Oceania and Australia, that form separate islands on the art network, meaning that you can be an artist who goes from one institution to the other one, and you have a lifetime full of exhibition opportunities to be part of the art world, but you have no path in this journey from here to the high-profile institutions in the center of the art world. We were able to use this data to predict even the future career of the artist. If you give me the first five exhibitions of a particular artist, depending on where he or she starts from, we can tell you where they're gonna get 20 years later. Why can we do that? We can do that because the art really works through a network effects. And these are very embedded in the way value is created in the art world, and this particular map captures that. What you see here is a work that is really a few weeks old, and I'm totally amazed that could be part of this exhibition. And what it represents is one of the hottest trends in the art space, which is NFTs, or non-fungible tokens. In March, the whole idea of NFTs in art have really exploded thanks to some very high-profile sales, which inspired us to start looking at the network effects of how NFTs have emerged. What is amazing about NFTs and generally crypto art recorded through these NFTs is that it's a totally open market. That we know who the artists are, and we know who is the first buyer. That means we know the primary market. We know how much they sold for, and we know how much they paid for those who purchased from the primary market, that the secondary market can be mapped out as well. And in this case, we created a map of one of the most highest profile NFT markets, so-called super rare. Each node here corresponds to an artwork that's sold on super rare in the last 3 years. That's the total length of the market itself. And two nodes are connected if during their lifetime they were owned by the same owner, that is, where the same collector has kind of had them. And when we mapped this out, we were puzzled by the existence of these very clear communities. And first we thought that these artworks that are all kind of seem to be owned by the same group of people, they're so connected, maybe they are connected because they are by the same artist. And that didn't really explain the effect. And then we realized maybe we should actually color this map based on who owns the work right now, based on the current collectors. And that suddenly made lots of order. So colors correspond to who is the current owner of the work. And it shows the presence of a couple of really massive collectors that really own this work. In many ways, this is no different from how a new ism emerges in the art world. We find that 87% of the artists, of the collectors buy the work, but they don't sell it again. So they hold on to it like many real world collectors do. And we also find that there are these large collectors, which, for example, are well known to exist, for example, even among Impressionist artists, who a few early collectors ended up purchasing many of their works and then reselling it to the larger community. In many ways, the art network that we saw earlier describes the traditional art market, where artists exhibit in museums and galleries and try to sell work. And what we see here is a 21st century, when the value and the valuation and the presentation of the work moved to the cyberspace. The question is, will this stay with us in the future, and how will that change the art world? Certainly, I'm glad that this could be part of this exhibition.